I open this academic ceremony in which Mr. Frédéric G.G. Dewez will defend his academic thesis entitled Spatial Omics to Quantitatively Study Tissue Heterogeneity. And I welcome, of course, the candidate and the panel, and also the promotion team, Professor Heren, Professor De Pauw, and Dr. Ballev, and also the members of the opposition who you will meet later on. Also welcome to family and friends here present and all the people online. But before we start the defense and the opposition, we will give the word to the candidate during the first 15 minutes to present his thesis for family and friends.
Thank you, Prorector. So, dear Prorector, dear members of the committee, dear family and dear friends, today is the final presentation of my PhD defense thesis titled Spatial Omics to Quantitatively Study Tissue Heterogeneity. So biological processes are those processes that are vital for an organism to live, to reproduce, and to interact with its environment. So these processes are carried out by millions of molecules and are organized in complex molecular networks that have a dimension in space and in time. So with the aim to obtain a comprehensive knowledge on these processes, there is a need, this requires large-scale analysis and therefore the use of high-throughput technologies. So amongst these high-throughput technologies, omics technologies have been developed. So omics refers to the Greek word entire, that means entire, and technologies refers to the study of the molecular class. So for instance, on this slide, you can see that the study of proteins will refer to proteomics, the study of DNA, genomics, and so on and so on. So there are equipments to study this class of molecules, and amongst these equipments, mass spectrometry has emerged as a really powerful tool to analyze biosamples. So what is mass spectrometry? So the principle of mass spectrometry is to physically separate molecules based on their mass to charge ratios. So by creating gas phase ions. What you obtain is what you can see on the screen. It's a mass spectrum, which is the plot of the intensity as a function to the M over Z. So the current MS standard technique is liquid chromatography mass spectrometry. So in order to analyze tissue sample, you first need to homogenize the samples because the study of biomolecules in their biochemical context implies complex mixture. So you first homogenize to first extract the molecules of interest, and then you have a second separation, which is in this case liquid chromatography. So this technique separates and sequentially elutes the different compounds of your sample before the ionization. So these techniques provide high sensitivity, high selectivity. You can study a broad range of molecules. This technique is an untargeted analysis. That means that you don't need an a priori on the molecule that you want to study. You can also obtain quantitative information. But due to the homogenization, you lose the spatial information, which is important to obtain deeper insight into these biological processes. So to directly obtain molecular information from tissue sample, MALDI MSI, MALDI mass spectrometry imaging has been developed. So MALDI means matrix-assisted laser desorption ionization. So you first apply a chemical compound which is called a matrix. So this matrix will absorb the energy of the laser, the energy of the laser, and this will be transferred to the molecules to create ions, so molecular ions. So what you obtain, as I said, it's a mass spectrum, and then this process is repeated in a grid-wise manner. So then for every ion, you are able to reconstruct an image and see the spatial distribution of this ion. So this technique provides high spatial information, so at the micrometer level. So this technique is also a non-destructive technique. That means that you can combine this technique to any other analytical technique. You can study a broad range of molecules. This is also a non-targeted analysis, but due to the direct analysis from tissue sample, you cannot add a separation step in between, and this leads to a lower sensitivity and no quantitative information. So combining these two techniques, so LCMS and MSI, so the analytical depth of LCMS and the molecular information from MSI would be a substantial tool to delve into the metabolome or the proteome or any kind of molecules. So one way to combine these two techniques is to use a laser macrodissection system, which is a light microscope equipped with a laser that enables to accurately cut out regions of interest. So therefore, I propose the spatial omics workflow where MSI is used to guide the laser macrodissection. So in more detail, what I've achieved is that you first analyze your tissue sample by MSI. Then this is followed by a segmentation that you obtain regions of interest that you can transfer to the laser macrodissection system. And in this case, then we could forward the samples to the LCMS to study the samples at the protein level. But to achieve this workflow, we needed to optimize every step of the workflow. So the first one was the image co-illustration. Indeed, with special information at the micrometer level, this co-illustration between the MSI and so from the MSI to the LMD needed to be really accurate. So we obtained this by the co-illustration of the MSI data to the high resolution optical image using the laser shots. Then we co-registered this optical image to the high resolution histological image using the fiducial marker. And then we could transfer this to the LMD using the same fiducial marker. So we achieved this co-illustration with an accuracy of 13 micrometer. This is in concordance with the daily spatial resolution used in MSI, which is between 20 and 50 micrometer. The second one was the image processing. Indeed, MSI creates really fine structure that makes the microdissection time-consuming and tedious. So we first, after the clustering, we perform a smoothing, then we remove the small object, we fill the holes, and then this enables the detection of the boundaries of the regions of interest that we upscale to the optical image resolution and then to the laser macrodissection system. The third one was the use of ultra-sensitive analytics. So with the help of Brucker and their newly mass spectrometer, so equipped with MALDI and ESI source, we were able to identify on average 2,500 proteins from minute amount of samples. So we decided to apply this workflow to study intratumor heterogeneity. So using the molecular information provided by MSI, we were able to, you are able to perform some statistical analysis that highlight tumor subpopulation that are not distinguishable with conventional histological technique. And these tumor subpopulations are important because they play a role in the outcome of the patients, including the tumor progression, the prognosis, or the response to chemotherapy. So we applied this workflow on the breast tumor section, where we first define the tumor area based on the histology. Then, based on the MSI information, we perform a clustering where we could identify 3 different tumor subpopulations. We transferred them to the LMD before macrodissection. We then analyzed the sample with LCMS at the protein level. So the proteome is dynamic and expressing and controlling the vast majority of the biological processes. Here, you can see that the biological processes were differently expressed between the different tumor subpopulations. So this workflow helps us to better characterize this tumor subpopulation. So to be more generic, what we have developed with this workflow, it's the study of the tissue section or tissue specimen using MAL-DMSI, or you could study a broad range of molecules. Then we could transfer this information to the laser microdissection, and then actually you could analyze the sample by any kind of omics technologies. While we focus on LCMS and proteins, actually you could also study the genomes or the genome or the transcriptome. So unlike LCMS that provides label 3 quantitative information, MAL-DMSI is a semi-quantitative technique. To obtain quantitative information from MSI, you need to use the current state-of-the-art approach. So in this approach, you have a dust tissue where you want to quantify the molecule of interest, so that could be a drug or a peptide. And then you have an adjacent tissue section, which is a blank tissue, where you spot a standard. The standard can be an analog or a stable isotope of your molecule, and then you are able to create a calibration curve. You apply then a label standard that is used to normalize the intensity. So then what you can do is you can map the intensity of your molecule of interest to this calibration curve to obtain the concentration of your targeted molecule. One drawback with this approach is the use of adjacent section. So depending on where you spot this standard, the signal might be different due to regional matrix effects. So like here, you can see that you have different part of the tissue, the mucosa, the submucosa, or the muscularis. So we developed a new quantitative approach. So in this approach, we created 3 different labels of our molecule of interest. We applied these labels in 3 different concentrations on top of our tissue. And then this enables to, at every measured position, to establish a calibration curve. And then we can calculate the absolute concentration of our target molecule at every pixel. So as proof of principle, we use the peptide histone H4. So we created 3 stable isotopes of this peptide. Here you can see the MSI image of this. So here the native H4 and here the 3 labels. Here on this image, we verified if the optimized concentration that we have applied on top of the tissue were covering the whole range intensity of this histone H4. Here you can see two examples of 2 different pixels taken into the predictive image, where you can see different slope and different slope of the calibration curve. And here, by visualizing the intercepts and the slope, we can see that the signal is affected by the morphological features of the tissue. And finally, we were able to calculate and predict the concentration of our peptide histone H4. So to validate our approach, we use the gold standard LCMS method. So I selected 3 different areas that I submitted to LCMS, and then I compared the absolute amount of histone H4 approximated by MAL-DMSI and by LCMS, and we could find a high agreement between the two techniques. So this was a validation of our technique. So to conclude, to study tissue heterogeneity, I developed a multivariate quantitative approach where we could quantify and visualize the spatial distribution of a wide range of molecules. This could help us to characterize the molecular changes in tissue microenvironment. And also, likewise, I developed an univariate approach where this is a more targeted approach where you could quantify a drug and visualize the spatial distribution in this drug, and this could help to run studies on pharmacokinetics or toxicity studies. So thank you for your attention.
Thank you very much, dear candidate, for a nice presentation. We will now start the opposition, and the opposition will be opened by Professor Honing, who's also the chair of the assessment committee, and he's professor of system imaging in the University of Maastricht. And today he has also accepted the role of secretary, for which I'm very grateful. Professor Honing.
Thank you, Pro-Rector. Dear candidate, first of all, a lot of congratulations for all your efforts to get desorption ionization technologies more quantitative. So this quest is really valuable, and it's great to see that you achieved a lot of results. But still, sometimes reading your thesis, I was a little bit bothered by the fact that you were using quite some semi-quantitative definitions and terms. So you're speaking on page 11 of the words analytical depth, and later on you use the words deep analytical insight. How deep and what is high? Because those are really semi-quantitative definitions.
So dear highly esteemed opponent, thank you for your nice word and thank you for your question. So it's true. So I will more compare, when I say deep, I will compare to the MALDI MSI approach. So if I compare both approaches, I would say that you can have a deeper insight into the, in this case, biological processes, or into the analytical, you can have an analytical depth, a better analytical depth with LCMS than MSI. But
so you mean a little bit more detailed information on the molecular structures?
So not maybe not the molecular structure as well, but also the in terms of numbers. So you can see the figures, page 33, where you can see the number of proteins detected by MSI and detected by LCMS. And you can see that with LCMS, you can detect way more proteins than with MSI.
I think you have a foreseeable character, because that's exactly one of the questions I had on my list. Because this figure 1.9 really disturbs me. Actually, what I read is that MSI is not able to see all this depth, analytical depth, while LCMS it is, even though I would argue that LCMS is also suffering from some lack of information. So where is MSI, in even quantitative MSI, bringing us compared to classical methods in getting this depth in analytical data?
So you mean the where is MSI quantitative compared to LCMS? Or?
Yeah, or I see that MSI is only able to see not even 10% of the molecules which are there. So what depth is there in mass spectrometry imaging, and will it deliver anyway?
So that's why we have developed this workflow. So we wanted to associate the MSI to the LCMS, because that's one of the drawbacks with the MSI. It's, as I said in my presentation, due to the direct analysis, and you don't have any separation, so you can have ion suppression, and this leads to like as I said in my presentation, lower sensitivity and no quantitative information.
So that's a perfect approach, because actually we are a multimodal imaging department. So one method is no method, maybe. And I would also like to discuss another semi-quantitative term you use, because I was a little bit puzzled on internal standards. Sometimes you speak about structural analogues or structural homologues, and then suddenly you would like to have an isotopic labeled molecule. So what's the difference between an analog and an homologue?
So an analog can differ from atoms or substructures, or also groups. And isotope, it's like in this case with the Stone Age 4, where we could have heavy atoms with arginine and so on. So yeah, the distinction, I'm not sure. Maybe can we call an isotope an analog, or I don't know.
Yeah, no, it's fine with me to use the definitions, but for example, what I was asking myself, what do you think are the criteria for an internal standard? And maybe out of the criteria, you will define what is an analog or a homologue. So
the criteria is that you need to have the same response for the molecule of interest and the analog. So in terms of ionization, in terms of, in this, if you use MALDI, in terms of ionization and desorption, so that's, you need to have you need to have the same, how can I say, the same response from the molecule of interest or in the analogs. That's
okay,
what
so you would agree they should have more or less the same physical chemical properties. Okay, then we are on the same page. That's very important. A very detailed small question. That's, I don't know the page anymore, but the figure is 1.12. Oh no, it is.
It's in the introduction.
It is on the introduction, and it's on the MS spectrum, I think. Yeah, it's on page 38.
Yes.
And you are speaking about sequencing of peptides. No, I completely understand what you're doing, but what is the actual power here? Because I miss all the B6 to B11 ions and fragments, and all the Y7 to Y10. So how sure are you with this picture that the sequence is your sequence?
So it's I agree. So we find actually we find so many different ions from the peptide, so that I think we can be sure that this is the peptide that we are looking, because I think we have like more than 10 more than 10 peptide ions from the peptide, so we can be sure that it's our peptide.
Okay, so you would say that by having Y1 to 6 and B1, but I miss B1, but B2 to B5 is sufficient from both sides to sequence this peptide.
To me, with all the ions that we have here, yes, I think we can be not 100% sure, but I guess we can be
pretty sure. Semi-quantitatively being pretty sure.
Exactly.
Yeah. Professor, one question?
Yeah, short question, short answer.
Just a small question. Proposition number 6, if someone can read that for me.
Proposition number 6, research means that you don't know but are willing to find out. Charles F. Kettering.
No, I support this in the sense that as a researcher you would always like to find out. But the beginning of this sentence states that actually you don't need to have any hypothesis of any idea. You just start somewhere and you find out. Yeah. And I hope that is not a normal academic approach in research.
No, yeah, it's true. You first need to have like the hypothesis. So you might have actually already gather some information, and then you just want to verify your hypothesis with the.
I think we completely agree.
Yeah. Thank you.
Thank you, Professor Honing. Then we come to the second opponent, who is also a member of the assessment committee, and that's Professor Eppe, who's online, and he's director of the Laboratory of Mass Spectrometry of the University of Liège. And thank you for coming all the way from Liège, albeit online. Professor Eppe.
Thank you, Mr. Prorector. Can you hear me correctly? Yeah? Yeah, okay. So first of all, I would like to congratulate Frédéric for the quality of the manuscript and the nice presentation of the overview of his results. So I'm going to focus on chapter 4, mainly on the quantitative approach. And the first question, it's a very nice and elegant approach. And the first question I would like to ask you is, when you prepare the sample, you have to assume that it's focused on peptide, on a native histone H4. How can you be sure that you have a very high efficiency in the digestion of the protein to give the peptide? And how we can, let's say, estimate the digestion yield in the imaging first?
Dear highly esteemed opponent, thank you for your nice words and thank you for your question. So I totally agree. So the digestion cannot be 100% total. So, but actually what we could quantify is only this peptide, so the peptide that has been digested, so from the protein. So actually what we have quantified is that the peptides coming from the digested protein. But we are not sure that all proteins from H2N4 were digested.
And what kind of technique can be used then to ensure that you have a high yield? To verify.
So to make sure that the all proteins are totally digested.
Yes.
Yeah, you could maybe label the protein before. So, but then you need animal model and so on, and then you could see you could then see the digestion and detect your molecule of interest and then quantify with our method.
Yeah. Okay. So now, if we move to pixel by pixel, so you use different labels, as you show different labels, compound, and also a different concentration, and then you were able to, let's say, calculate a slope pixel by pixel and take that into account. Let's say in situ for in situ quantification. So I would like to know what is the extent of variability of the slope pixel by pixel. Can you comment on that? Is it specific region or subregions? And also, what is the reason? How can you explain that we can have such a variability of slope pixel by pixel?
So yeah, we had more variation with our method than compared to the single label method, because so for the single label method, you compare the signal and you map the intensity to only one calibration curve. And in this case, we had several calibration curves at every pixel. So this I agree. And the different slope comes from different response signal from the tissue, due to the morphological features of the tissue, or just because of chemical interaction at this specific pixel compared to another region. It might be, it can also be ion suppression at this specific position, and not this specific position, but these pixels, and compared to another area where there were less maybe cells and less ion suppression. So that's why the variations of these slopes are in this method.
Yeah, yeah, correct. And then this is very elegant approach, but it's what we can call a targeted mass spectrometry imaging. And you show that with this specific example, it can work. So, but in your introduction, you mentioned that mass spectrometry imaging is a non-targeted approach. So I would like to have your opinion how we can extend this approach, let's say for other peptides or other interesting molecule to monitor in mass spectrometry imaging. What is your opinion about that?
Yeah, yeah, that's true that my approach was really targeted to only one peptide, and we needed to prepare the isotopes based on one our molecule of interest, so based on one peptide. So actually, at that moment, I don't see a way to have quantitative information of different peptides. No.
No, it's really targeted. Yeah, but this response factor for that peptide with these homologs and with this label compound cannot be extended to other peptide of interest.
The thing is, as we said with Professor Honing, it's that the peptide, like the internal standard, needs to have an analog structure. So yeah, it needs to have the same physical properties and physical-chemical properties than the target molecule. So in the case of peptides, you have so many amino acids and so many different interactions that I don't know if you could, I'm not sure that you could use this, for example, peptide histone 4 and the label that we have created to quantify another one. So I'm not sure that this is possible.
Okay. Then I have time for one last question. Yeah. Yeah. Thank you. So in the lab, you have also a MALDI2. What do you think that could be interesting or not to use MALDI2 for MALDI2 for the peptide analysis in mass spectrometry imaging or not?
With the quantitative study or for?
Yeah, for quantitative, for efficiency of ionization process first for peptide and second for quantitative approach. Does it mean that it could be an added value or not?
I'm not sure that it will be an added value. Yeah, I'm not sure it will be an added value for peptide, but if you want to quantify small molecules, for sure. So this will help to better ionize your molecule of interest, and then you will have a better vision, a better view of the molecule of interest that you are quantifying. But I'm not sure.
Yeah, you mentioned small molecules, and what families of compounds you have in mind?
So in our lab, we also study lipids, but it also can be different drugs. So also in our lab, actually my PARANAF showed that MALDI2 could help to better ionize some different drugs. So I think this could help to better quantify the drugs of interest from tissue section.
Okay, thank you very much.
Yeah, you're welcome.
Thank you, Professor Eppe, for your opposition. Then we come to the following opponent, who's also a member of the assessment committee, and that's Dr. Heijs. He's working in the Center for Proteomics and Metabolomics in the University Medical Center in Leiden. And thank you, Dr. Heijs, for coming all the way from Leiden so early in the day. Thank you very much, and the floor is yours.
Thank you, Mr. Prorector, and congratulations, Mr. Candidate, on a wonderful thesis. I read it with great pleasure. I do have some questions, and it actually starts in the prologue on page 13, because you state there this dual approach of using MALDI MSI and LCMS requires optimal instrumentation on both parts. So was the instrumentation that you used in your chapter 2, was it optimal for doing the task?
So, dear esteemed opponent, thank you for your nice words and thank you for your question. So for chapter 2, you mean? To the chapter 2 we
sorry, the chapter 3, so the first approach for the co-registration chapter, basically.
So yeah, actually, in this chapter, what we use the Synapse G2SI for the MALDI and 2D Q Executive, 2D LCMS and Q Executive for the LCMS. So yeah, the Synapse is maybe not the yeah. How can I? No, I'm not. I can't say that. At that moment, the Synapse was the instrument available that were able to analyze PN membrane slide. So this was needed for the microdissection, yeah. Which is not possible, for instance, with the Rapuflex from
so it was fit for purpose at the time.
So exactly. Perfect. So for this, it was so at that moment it was the perfect instrument for this approach, and the 2D LCMS was developed in Liège, where they have their it's their how can I say that. They developed this technique for years and years, so they have definitely optimized every step of the LCMS and also the sample preparation of the LC and the instrument.
So I have a follow-up to that question, because in the next chapter, you describe the same measurement, MALDI MSI and LCMS, on a single instrument. So can a single instrument be optimal for both, or will you always have a trade-off?
So actually, we had really, so for the MSI part, I would say this instrument would help to get higher spatial resolution first of all. So you can go up to 5 micrometer, while before it was only 15. So with the synapse, and we also had higher sensitivity with this new mass spectrometer. For the LCMS, it's absolutely 2 different approaches. So for the first chapter, it was you had first a 2D LCMS, so a 2-dimension separation, and then followed by the Q-exactive. But in this case, they developed a new method, which is the so in this mass spec you have the TIMS funnel. So I don't know if it's scientifically correct, but in my mind, it's also a 2D separation between the analysis. So you have first, because you have first the LCMS, and then you have a second separation, which is in this case the ion mobility. So in this, I think the second approach with this instrument will help to actually develop this workflow, because a single instrument it's much easier to handle than 2 different ones.
I can agree to that. Okay. So I also would like to focus a bit on your quantification chapter 4, I think it is.
Yeah.
So I checked the code that you use to perform the quantification, and what I noticed is that you only use monoisotopic information from the protonated molecular species that you detect for your peptides and your standards. So in my mind, the amount of isotopes that you get tells you something about sensitivity and thus quantification, and also the presence of adducts, sodium potassium adducts can change throughout the tissue. So can you comment on why you chose to only use monoisotopic peaks?
Yeah, thanks for your question. I agree that we only selected the monoisotope ion, and that's true that if we had adducts, they were not quantified because they were not taken into account in our. So this might be an improvement that we could apply in the future in the case of if you use peptides in this case.
Do you think you can get better correlation to the LCMS data that with the inclusion of isotopic information? And
I think so. Yeah, I think so.
Okay.
I would say yes. Yeah.
Interesting. And then if there's time for one more question. Then I also have a question on the co-registration that you performed. I think it's a quite clever strategy that you use to take a high-resolution optical scan of the analyzed tissue to see the laser marks. And I was wondering whether taking this approach will not limit your study. So can you maybe comment on how this approach would still be working or not if you use, for example, a continuous laser rather than a pulsed one, or maybe a DESI source where you don't even have laser marks?
So this is actually, you pointed out, it's the drawback of this approach. So if you don't see the laser shots using this instrument, it's the co-registration will be less accurate, because we decided to use this approach because the software of this instrument doesn't enable to obtain the transformation matrix and the co-registration numbers. So that's why we had to use this approach. But for instance, when you use the Bruker instrument, you can directly co-register the optical image using the fiducial marker.
Mm-hmm.
And in this case, you don't need the laser shots anymore. And then you don't need to see the laser shots anymore. So in this case, yeah, it was really specific to one instrument, to the use of the synapse. And I think if you use any other kind of instrument, this co-registration might not be the best, or might not be used.
But you mentioned in your discussion that by using a higher magnification, you can get a higher accuracy, right? So what does that mean if you want to go to single cell resolution? Is the higher magnification microscope, is that going to be enough, or do you need more?
So I think I don't remember the numbers exactly, but so there are many ways to improve this workflow. So we, as I said, the first construction was between the MSI and the optical image using the laser shots. And then by using a higher image resolution for this image, you will have already a better co-illustration for sure. Then the second was with the histological image, the same. If you have a higher image resolution, for sure you will have a better co-illustration. And then the third one was into the LMD. And then in this case, we use the 5 times magnification. And it's a light microscope, so we could go up to 64 times. So this will definitely help for the co-illustration. And then this will help to see the single cell from the tissue.
But are there any other limitations when it comes to using a higher because it sounds too easy to me to just use a higher magnification and then it works.
So yeah, there are several limitations. So the first one is already with the LMD, you are limited to the field of view. So if you import regions of interest and they are bigger than the field of view, you cannot cut out this region. So this is already one limitation. And you also need to have higher mass spec imaging resolutions, because in my case, I used 50 for the chapter 3. And of course, if you want to go to single cell, you need to go lower in spatial resolution. So you have like every step of the co-illustration need to be optimized in the case of the study of single cell.
Thank you very much. I can work back to that.
Thank you, Dr. Heijs, for your opposition. Now we come to the following opponent, who's also a member of the assessment committee, and that's Professor Darmink. And he's Professor of Translational Research in Gastrointestinal Surgery in the Maastricht University Medical Center and the University of Maastricht. Professor Darmink.
Thank you, Mr. Prorector. Dear candidate and your team, I really, really want to congratulate you, because we had long discussions in the merger with, or the collaboration between the surgical department and the M4I department, and how can we use it in the clinic. And there to really use it, I think we need to embark on the steps that you take, is to have a quantitative MSI. So I really enjoyed reading it, and I really enjoyed the steps that you made. I would like to start with a very short and probably easy question, and it's in page 83, and it's figure 4.6. And there you compare 2 methods, and then you state that the agreement you test by a Pearson correlation coefficient. Why did you use Pearson?
So dear highly esteemed opponent, thank you for your nice word and thank you for the question. So can you repeat the page?
83. It's the right panel.
Which one?
The right
83.
panel.
Okay, yeah, sorry.
Yeah,
So
4.6. So there you show the absolute amount of histone, either by the MALDI QMSI or by the LCMS technique.
So what we did here is that we first performed the MALDI MSI and the contact.
Just these statistics. So why did you use?
Why? So we wanted to, in this case, find the correlation between the two techniques.
So if you use Pearson and you have 2 different methods, but they are on a systematic error different from each other, there are 2 parallel lines, and the Pearson will be 1. But that's not what you want to show, is it?
No, in this case, it was only region by region. So the absolute amount of estimated by the MALDI and the LC.
It's an indirect approach to tell you that Pearson can give a misjudgment. I think you should use interclass correlation, or maybe even go for a correlation variation or a Blend-Eltman. Why do you think I would go for a Blend-Eltman comparison with 2 methods?
So I don't know this.
in a blend-Altman correlation. So it really has your gold standard or a different method. And the differences is expressed in the quantitative amounts. And because it's so neat that you do quantitative, this gives a amount for the reader. So for us, an insight about the change that you can measure between the two methods. So maybe you should think about it and do after the party of today. So I would like to go to the propositions, and then I would ask you or one of your paranymphen to read proposition 5.
The investigation of intratumor heterogeneity is of utmost interest for the scientific community to better understand the evolution of cancer in this thesis.
So I completely agree, because this heterogeneity is really a pain in the dot for clinicians like me. So if you give chemotherapy, some part of the tumor reacts and some part doesn't. So let's do your postdoc, and I hire you. Let's do an experiment in which you're going to tell me how I can show, either by the MALDI guided approach that you describe, so the laser dissection, or with the novel targeted approach that you showed in chapter 4. So you're going to show me what the differences in protein synthesis between the tumor, and then to make it really easy, we go for organoids. So not in a tumor in a human, not in a tumor in an experimental animal, but just the easy organoids that have their heterogeneity and the chronicity. So I think that's as easy as can. Which of the two methods and how are we going to do it?
So actually, we could analyze the proteins, but we could also analyze any kind of molecules in the LCMS part. For instance, if I use the spatial omics workflow, so actually we could obtain a really broader view of all the different kind of molecules that you could have in your organoids in this case. So I think I would go with this approach, with the spatial omics workflow.
Which one? The
spatial omics workflow. The MALDI MSI followed by LCMS. But then they are so small. I know that the organoids are really, really small. And this is really challenging first for the MSI and then also for the LCMS in this case, because what we had in this thesis, it's the tissue sample, and the LCMS was optimized for only 2,000, for one region, 2,000 cells. So I think organoids are smaller than this. Huh?
But the other one is targeted, so you can really go into low enrichments.
You mean with which method?
With your fourth method, in which you have your isotopic 3 different. So that in this case, might be very interesting too, isn't it?
Yeah, this also too, because then you don't have the LCMS and you have only the MSI, where we could quantify, I don't know, maybe the chemotherapy that you've given to the patient. Well, not to the patient, but in this case,
you've got a targeted peptide that you look at that doesn't grow when it reacts to chemotherapy.
Yeah.
So which is the best?
I would go for maybe the second one then, because
I think it's a postdoc question, isn't it?
Yeah.
So you should
maybe you could combine both, and then you are
no, will remain open. One final question, and that is proposition 7. So maybe the other paranymph can read proposition 7.
7. One of the ways to learn is to know when you are making failures. Robert again.
So what was the biggest failure you made in the last 4 years, and what did you learn?
I don't have enough time to to talk about all the failures I had during my PhD. So, but I had I can remember only the first one was just to get to understand how the regions of interest can be imported into the LMD. This was already a big one, a hard one. And then there are so many, I can't even mention all of them. It's like, even for the quantitation, it was really because it was really a novel approach. So we really needed to think of all the parameters and all the conditions that we used in this chapter. So the failures, I think Ron and Benjamin can answer your question by saying a lot of failures. Yeah.
Okay, please with the answers. Give
Thanks.
the word back to the prorector.
Thank you, Professor Darmink. Then we come to the following opponent, and that is Dr. Freykin. He is Associate Professor in Quantitative Imaging in Mass Spectrometry in Institute M4I in the University of Maastricht. Dr. Freykin.
Thank you, Mr. Prorector. Dear candidate, thanks for be present here. I want to congratulate you with the nice work you delivered after a couple of years hard work with a lot of failures. I heard. I'll come back to it in a minute. So and also congratulate your promoter and your co-promoters in achieving this work. So interesting thesis I mentioned already. There are several points where I'd like to address later some time to. But first, in follow-up of the last discussion about the number of failures, I would like to have one of your paronyms read proposition 8, please.
So it's going to be in French. Produire beaucoup, ne publier que le meilleur. So in English, it means produce a lot, publish only the best. Jules Renard.
Thank you. So coming back to science, we are reporting the best. Are we not entitled also to report on the failures we make and what doesn't work? Because here you state we're not going to report anything of that.
Dear highly esteemed opponent, thank you for your nice word first, and then thank you for your question. So I agree that the failures can be also important in the analysis of the problem, and this can also help the scientific community if you are reporting the failures. But I don't know any papers that only report failures. So
I'm not referring to only failures. Of course, you can add the failures to the right
exactly.
conditions. So
yeah, I think it could be interesting to also report failures and.
It's not interesting. Should we not be entitled? That's my point. As a scientist, as getting this degree.
But then the publication will be too long to me because I don't know. Entitled, why not? But I'm not sure that it could be relevant. But I don't think. I don't know. I don't see how to answer this question. Sorry.
Okay. So we are in disagreement here then a little bit because you're stating it really boldly.
No, because failures are important, but are also important for the person that are doing the experiment. So to also understand what had worked or what hadn't worked and so on. So I don't know if I make a failure, maybe this would not have been the case for another. So I don't know if there is a need to report this failure in this.
No, that can come from the scientific community if it's a really valuable failure. But anyhow, let's move on. And I'd like one of your paronyms to read proposition 2 for me, please.
Proposition number 2. Mass spectrometry imaging continues its development towards high spatial resolution and cellular sensitivity, but still lacks chromatographic separation to eliminate ion suppression. This thesis.
Yes, touching on a very important point, which was already discussed several times, ion suppression. So how are we going to tackle it now? Because this is an open door, right?
For MSI, you mean?
Yeah.
How to tackle that? So we have already talked about it, but you can also use the MALDI2. So,
does it work always?
Not always, not on all the drugs. So this is also this might be different depending on the molecule that you are studying. Then you have different ways to improve this. For instance, if you want to study peptides or proteins, you first need to have some washing steps that you can use before to remove some salt and some lipids, some small molecules to decrease this ion suppression. And what else? I think I don't see for now. Yeah, that's because of the direct analysis from tissue section. This makes this is difficult to add a separation step in between.
Yeah.
So,
okay, so we'll leave it with that, because I think we can talk about this several hours on all the possibilities which are there and which have not or have been applied. I'd like to move to a very small question on page 60, where you state, let me quickly go. It's the first line, and if you have to read the whole sentence from the previous page. Although the LMD is able to dissect single cells, the vast number of small objects encountered makes a dissection infeasible. So how does that work? So I'm able to dissect single cells, but if we have multiple single cells, we cannot do it.
It's not infeasible, it's just time-consuming and tedious. So in this case, first of all, the spatial resolution was not not the goal one to study single cells, so there was no need to microdissect single cells. So in this case, we could remove the small objects to make the microdissection more feasible. Like in this way, less tedious and less time consuming. So
yeah. Okay. Thank you.
That's it.
Then I'd like to move to a more often discussed chapter 4 already, quantitative approaches. And I've got several questions there, but one of the main questions which I have there is comes back to the statements around page 82, where you're talking about the multi-label, single label experiments and methods. Being in the group for a couple of years, if you've also been, you had some colleagues, former colleagues like Lieke Lamont and Leonard Huizing. They also worked on approaches for getting proper quantitation. So what I want to know from you now, after having worked on this multi-label approach, what are the pros and cons of all the three methods? Like what Leonard be using, he'd be using a single label approach. Lieke Lamont has been working on tissue homogenates, spiking those with a calibration line. And you're working with the three-label approach. And then when you compare those ones, how do they compare with throughput, sensitivity, dynamic range? What's going to be finally deployed in a more robust or industrial environment?
So, for instance, my approach, where you apply the internal standard on top of the tissue, for instance, this doesn't take into account the mass transfer kinetic energy. So it means that if your molecule of interest, depending on where the molecule is in the tissue, so close to the bounded to the walls and so on, so this might have an effect on the signal response. So in this case, this is actually a bad effect, and in the case of homogenates, so you don't have this because everything has been homogenized, and then you can spike your internal standard. But how to make sure that the internal standard is entirely in your homogenates, like properly distributed in your homogenates. So this also can be because then if you have a tissue section from this homogenization tissue, how can you make sure that your internal standard was well distributed in your homogenates?
Oh, that's grinding and these kind of things and homogenizing, so that can be done. But the point, of course, is that you're diluting all kinds of factors with your homogenates, which you're not picking up anymore. So, okay. So let's move to an intriguing statement you're making on page 83 on figure 4, just above figure 4.6. Where we're looking at this comparison between these two methods, the MALDI QMSI and the LCMS. And you state there, they revealed a high agreement between both approaches. This shows the high recovery of the target. But having a high agreement doesn't result in a high recovery, to my opinion. That's what you're stating here. So what actually do you mean? Are you really talking about recovery here?
So yeah, what we mean by our this high recovery, it means that by the
Okay, yeah, the answer has to wait, I'm
sorry,
afraid.
I will answer during the restaurant.
Over lunch.
Yeah.
Thank you, Dr. Freykin, for your position, and Mr. Candidate, Mr. Dewez. The time appointed for the defense of your thesis now has passed, and the degree committee will withdraw to another room to discuss the quality of your thesis and of your defense. And I request that you and your company await the results of our deliberations and our return in this room.
Hey, yeah, thanks. Okay. Tu restes debout, bon, faut pas.
I reopen this academic session. Mr. Candidate, Mr. Dewez, the degree committee here present has assessed the quality of your thesis and of your defense, and in view of its positive verdict and taken into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. And Professor Heren is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor. En blijven zitten.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes.
Then, by the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Frédéric G.G, the degree of doctor, and grant you all the rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by Rector, the Secretary, and the other members of the committee and affixed with the official seal of the university. Dear Dr. Dewey, dear Frédéric, dear Fred, congratulations. It's done. I'm glad that I'm the first one to congratulate you with this newly obtained honor. Today is Thanksgiving in the US, a day of giving thanks and sacrifice for the blessing of the harvest of the preceding year. A very appropriate day to finish a PhD. In your case, an almost 5-year journey that was always on the edge. I'm using the term on the edge proverbially, as many aspects of your PhD were on an interface between two largely varying elements. And we've heard a lot of these elements already discussed today. You started in December 2016 at M4I as one of the last of the French contingent in a project in the framework of what was then called Imaging Valley, a collaborative project between the University of Maastricht and the University of Liège, defined together with Benjamin Balloff, your co-promoter, and Edwin de Pauw, your other promoter, and present here today. Edwin, you are online, but I would like to thank you and your colleagues for the fruitful collaboration between our two institutes in this beautiful Euregio. And Gauthier, I hope there's more to come, and I thank you for your presence here today and your thoughtful comments to Fred's thesis. A valley, and I guess in this case the Meuse Valley, is defined by two edges. In between, things happen. And in your case, Fred, you made the science between the two institutes happen, and for which all of that we are very grateful. Obtaining a PhD is by definition a learning process that explores and challenges the limits of what you know. And failure is a big part of that learning process, as you very carefully commented on in one of your propositions. You are trained as an independent researcher, and that is a skill that you do not possess immediately after completing a university education, but one that you have to learn. A skill that actually stands in stark contrast to the collaboration in a research team that you have done in the recent years. Working in a team limits your scientific freedom to some extent, and John Simon writes about this in his book, The Force of Knowledge, and he writes, and I quote, after I turn the page. How can a scientist in training in a team internalize the values of scientific imagination and criticism and the balance of radical propositions and conservative skepticism? After all those are crucial qualities of the scientific mind. You have proven in your work how you can achieve a scientific achievement in different teams and collaborations. Learn a lot, and without giving up your scientific freedom and skepticism. You and your supervisor Benjamin definitely do not like that. Certainly not in the early days of the new approaches that didn't work, instrumentation and analysis tools that were definitely not yet performing optimally. The challenge of this project was that you also moved along the edge of our joint abilities. You started to explore a unique combination of imaging mass spectrometry and proteomics to study the heterogeneity of cancer in very different ways. Doing research in this setting can only mean that you don't know yet, but are willing to find out. And that is what you did very successfully in these years. You've managed to produce 8 papers, a patent, and a thesis, which is in my mind a testimony of your drive and willingness to find out. But maybe more importantly, while moving to the edge of science, you turned out to be a real team player. Half of your publications form the heart of your dissertation, but it's precisely the other ones that didn't make it into your thesis that indicate how important your role has been in the work of others. With these papers, you show what kind of team player you are in science, whether it's Britt, when she was still an intern, with Florian, with Janwa or Clara, or many of the others that you collaborated with. This quality you should cherish. With the increasing interdisciplinarity of science and industry, that will often come in handy in the future. Together with the Liège team, you integrated imaging and omics, 2 vastly different fields, or maybe not, using the same analytical tool, a mass spectrometer. During your PhD, you managed many different mass spec-based approaches to really unravel the complexity of biological systems. It was the basis of the spatial omics approach that you later on developed in close collaboration with our friends and colleagues at Bruker, and resulted in some very nice instruments and approaches being installed at MFRI. Emma's guided proteomics with a laser capture microdissection step in between. An approach that we further developed, and many of your successors will be using it. But remember that you laid the foundation for all that work to come. The field is now moving heavily towards even single-cell spatial omics, and we already touched upon that today. And you started that in the collaboration with Clara that made it into Angewandte Chemie. You helped to develop and improve the single-cell precise co-registration. Also, again, at the edge of our knowledge and at the interface of 2 disciplines, MSI and bioinformatics. So much for the science. Now let's talk about the French connection. I'm actually glad to see many of them here, and to see a team of friends, your paronyms now, here in front of me, all graduates from MFRI, is actually a pleasure for me to witness as well. The French connection also has adopted some Chinese friends, but many of them have now moved new positions, mainly back in Lille. It has always been a pleasure to see how you socially interacted with the other researchers at MFRI. It is these types of social interactions and cohesion that we now miss dearly in this pandemic. I want to thank you for your friendship throughout these years and everything you've done to make the group even more social than it already was when you joined. I will say, though, that the Dutch culture has rubbed off on you a little bit. Many of the committee members here did not receive their printed booklet for the thesis, and rumor has it it was cheaper that way. That sounds a little Dutch indeed. All right. One of your propositions that you are a sore loser. One of your propositions was that you are a sore loser and only play to win. I could not help but to conclude that you were talking about research. And maybe that's not true. But there has been a lot of debate in the Netherlands recently comparing an academic career with top athletes, top sports, doing both requiring time, energy, effort, creativity, discipline to result in success. Some people will argue that both need a lot of effort that only goes into small improvements and that often does not get the recognition it deserves and requires significant self-sacrifice. You already talked about failures. There's, however, a major difficulty with these sports metaphors. They forgo one thing, and that is the unpredictable nature of science and the curiosity that drives a good scientist like yourself. In top sports, you do one thing extremely well. In science, you have to do extremely well in many things that continuously change and do not work. Looking at it that way, the science-sport comparison might be less motivating than intended. And there's actually a study that demonstrates some evidence to that effect. I personally believe that playing the game of science in the last 5 years has brought you the fun and excitement, and of course, today's win. I want to end by saying that I'm thankful for everything that you have realized at M4I and the way you contributed to the innovations and success of the Institute. I will hand you the M4I medal later today that depicts Prometheus carrying the torch of knowledge as a token of our appreciation and to celebrate your success. I hope you will keep on carrying this torch as a team player along the edges of science throughout your career. Congratulations once again on your newly obtained honor. And of course, I want to include your partner and your extended family in my congratulations. I'm sure that they are all as proud as I am of your outstanding achievement here today.
Thank you very much.
Dear Dr. Dewez, also on behalf of the Board of Deans of the University of Maastricht, I would like to congratulate you with the honor you have acquired. You've seen a great promotion and you did well. I also congratulate, of course, your partner, family, and friends, and all people online, all your whole network. And of course, your supervisors, Professor Heren, Professor De Pauw, and Dr. Ballev. I hope you have the chance to celebrate it today. In these Corona times, it's difficult, but take the time for that. It's an important day. Thanks to the Pedel and Luc Peters for their support, which made it possible for everybody to see this online. And thanks to the members of the opposition, of course, for their opposition and the judging of the manuscript. And having said that, I wish everyone a pleasant day, and I close this meeting. That means that